Hello guys, welcome to Easy Bio. Today's video is going to talk a little bit about genetic engineering. I follow National Scottish Curriculum and I have a playlist dedicated to that, so you can go and check that out if you want. Okay, so let's focus now on genetic engineering. What is genetic engineering? Genetic engineering is changing the material of an organism by using various different methods and techniques for the organism to have desired characteristics or outcomes. Before we progress any further, let's learn what genotypes and phenotypes are. They will be important a little now, but definitely later. A genotype is simply the combination of genes an organism has inherited. And a phenotype is your physical characteristics. So things like hair color or eye color. We'll talk a little more about genotypes and phenotypes in another video, but if you want to find out more right now, I'll leave a link in the description box for you. Okay, back to genetic modification. So, how genetic modification works is that it gives an organism, remember, an organism is a living thing that can survive on its own, a genotype that will change its phenotype so that it will do something to the organism to alter the combination of genes. This in turn causes the physical characteristics of the organism to change too. Why do we genetically modify living organisms? Well, that's because we can alter their phenotype in a way that suits us. For example, making it bigger or making it last longer. A lot of the food you eat has probably been genetically modified. Potatoes, tomatoes, milk, and rice are usually genetically modified. But what we're going to focus on is genetically modified bacterial cells. Usually, bacterial cells produce something called toxins, but by genetically modifying them, we can make them produce insulin. Insulin is basically a hormone that lowers your blood sugar levels, and so it's really important. Type 1 diabetics have problems with producing insulin, and so they need to be artificially given insulin. This is where genetic modification comes into play. So, how do we get to this stage of bacterial cells being able to produce insulin? Well, first, the place on the DNA that codes to make insulin has to be identified. Then, a specific enzyme called the restriction enzyme is used to take the gene from the human chromosome. If you remember from my cells video, and if you haven't watched that, please go and watch it to gain a better understanding of cells and what I'm talking about. Basically, bacterial cells have small, round forms of DNA called plasmids. These plasmids are then removed from the bacterial cells. The DNA of the plasmids are cut using, again, a restriction enzyme. Then the human insulin gene is put inside every plasmid. So the plasmid is basically a vector. A vector is something that is used to transfer a gene from one organism to another. The genetically modified plasmids are put back inside the bacterial cells. With that, the bacterial cells will be able to produce insulin since they have the gene to produce insulin. When the bacterial cell reproduces and replicates, a lot of insulin can be made. This diagram is a really good representation of the entire process. So if you still have a little difficulty understanding the stages, this could definitely help you. But anyway, that's the end of the video. If you have any problems understanding genetic engineering, post them in the comments and I will definitely do my best to answer them in a way that you can understand. My next video will be about respiration, so I will see you then. Bye.